Hi, I'm Alex, and my PhD research has focused on a meta-analysis of genomic datasets for the purpose of identifying high-confidence transcription regulator target interactions. And so we hope that this can be used to provide positive labels for predictive methods, to learn more about the underlying biases of these methodologies, and to hopefully identify biologically meaningful and underexplored regulatory interactions. So the first leg of this research just got accepted into genome research. ID is still pending, but basically I created a workflow for the identification, aggregation, and joint analyses of TR ChIP-seq and perturbation data. And these are two of the main methodologies that test direct regulatory relationships at a genomic scale. And so we focused on a subset of 8 regulators of importance in both mouse and human, looking to exhaustively identify the data for both species. Okay, so the ultimate purpose of this work was essentially to rank target genes. However, we wanted to better understand the contributing features of these methods. So in the paper, we look at things like the similarity of the experiments, generic signals, so gene targets that are prioritized regardless of which TR or context is assayed. We develop a mixed effect model to look for specific binding for a given TR relative to the others. And then we also look at how well these aggregated genomic lines of evidence can recover literature curated targets. So these are independent sources of information from low throughput biochemical assays. But again, the primary focus was to prioritize gene targets for each regulator. And so here I'm showing as an example for ASCL1 for the ChIP-seq. Each column is an experiment, and this final column represents the average binding score across experiments. So we can see examples of genes that have strong evidence, weaker to moderate evidence, and essentially no evidence. Similarly, for the perturbation data, each column is an experiment, and then again, we see this gradient of evidence. And so we combine this information by taking the rank product of these two aggregated lists to get an integrated genomic ranking. And so here I'm showing each point is a gene. Those in blue have literature curated evidence, and those in gold have both literature evidence as well as a high integrated genomic ranking. Okay, so for work that's currently in progress, we want to see how well single cell RNA-seq data can recover these regulatory interactions. And so obviously what I've just been talking about involves a lot of aggregating across biological contexts. And so this collection of data will also help us to kind of get better granularity of in which cell types these regulators and candidate targets are actually mutually expressed. However, the primary focus, again, is to look for reproducible signals. And so to do this, we're leveraging existing work that builds single cell co-expression networks and aggregates them to kind of strengthen the evidence that they provide. And so I'm still working on things like understanding the effect of the count matrix processing on some of this output, but the underlying idea is quite simple. For a given data set, for each cell type, you construct a gene-gene correlation matrix or network. You rank standardize these edges, and then you sum them over each cell type within a data set. So this gives you a final aggregate network from which you can extract a single vector corresponding to a transcription regulator. And then this represents the ordered list of genes that whose transcripts co-vary with that regulator after correcting for cell type compositional effects. And so this can obviously be done in a cell type specific manner, which is what Harris 2021 did for neuronal cell types. We'll look at this depending upon the consistency of cell type labels, but right now the primary focus is looking for consistent signals across cell types and data sets. Again, the underlying assumption being that this speaks to biological importance, while we'll also be cognizant of the potential biases that come from this kind of aggregation approach. So then we'll take these lines of evidence, these ordered target vectors, and then we'll see how well they're able to recover the genomics and low throughput evidence. Um, and so, here I'm showing a pretty optimistic schematic of what a precision recall curve would look like in that sense. Based upon our experiences and the literature, we do not expect this overlap to be that strong, but we do expect it to be better than random, and that this speaks to the underlying biological signal of regulation. Other questions we're hoping to address are things like, can we identify repressor relationships, which are inherently more difficult to identify than activation? So this will be done by looking at relationships that are trend towards negative correlations in combination while still having binding evidence, as well as the sign of the fold change relative to the perturbation type. And then finally, we'd also like to identify potentially conserved interactions. The selection of TRs we've looked at are highly conserved in mouse and human. They have similar tissue expression patterns, and mutations in the TR also lead to similar phenotypes. So we would like to identify candidate targets that might be conserved between the two species. Okay, thanks for paying attention.